I think we will start. I hereby open this academic ceremony in which Miriam Fichtner will defend the academic thesis Features of Muscle-Specific Tyrosine Kinase Autoantibodies and B cells derived from myasthenia gravis patients. May I invite you to present a summary of your study and the conclusions of your thesis.
I want to thank the ProRec very much, a lot for this kind introduction, and will now proceed with the presentation part of my defense today. Can you guys verify that you can see the slides? Perfect, thank you.
We can.
Perfect. Ladies and gentlemen, highly esteemed members of the examination committee, fellow scientists, and distinguished guests, I'm honored to stand before you today to defend my doctoral thesis at Maastricht University. This moment is the culmination of many years of rigorous research, dedication, and the unyielding commitment to the pursuit of knowledge in the field of neuroimmunology. Before I delve now deeper into the details of my thesis, I want to express my sincere gratitude towards my advisors, my mentors, colleagues, friends, and families, without whose unwavering support and guidance, I would not have been able to accomplish this. So thank you really very, very much for everything. So, as I now embark on the journey of defending my thesis, I'm reminded of the words of Albert Einstein. The most beautiful thing we can experience is the mysterious. It is the source of all true art and science. And indeed, the pursuit and understanding of the mysterious guided my PhD project, and for those who know me personally, is the essence of me as a human being. Today, I will tell you about the mysteries that I explored, the questions that I thought to answer, and the impact that my research has made to the field of myasthenia gravis research. Myasthenia gravis is a disease that I focus on in my research. It is a rare disease where the communication between nerves and muscles is affected. Patients due to that experience muscle weakness and increased fatigability. And this debilitating disease is caused by autoantibodies. These autoantibodies act like roadblocks that specifically interfere with this interaction. And in chapter 1 of this dissertation, I focused on solving mysteries surrounding these autoantibodies and understanding these better. So first now, we will try to understand better what antibodies are and how they function. Our immune system produces antibodies as part of our defense against foreign invaders, such as bacteria and viruses. And they work as a key lock principle. That means they act like keys that specifically bind and recognize the locks on these foreign invaders. And this interaction actually is highly specific. Unfortunately, in diseases like myasthenia gravis, there's autoimmunity involved. What does that mean? These keys, these antibodies, mistakenly detect locks on our own body cells and our own tissue. This is very important for the research that was done in chapter 1. For further research, it's also very important to understand that B cells are the ones that produce the antibodies, and that the B cell receptor or autoantibodies or antibodies are also present on the cell surface of these B cells. And as you remember from the slide before, this interaction is highly unique. So we can use this uniqueness to track and identify and trace autoantibodies and B cells. And that was done for chapter 2 and 3 of this thesis. Now it remains unknown to people from my family side that now listen with how can I as a scientist access B cells and autoantibodies. When a patient donates blood, we are able to separate everything into different fractions, and we are able to separate antibodies from B cells. Also, this is already a very good approach, and we can investigate B cells and antibodies individually. There are still many limitations that are associated with this approach. One limitation is that the antibodies in serum are highly heterogeneous. We cannot attribute functional mechanisms that we find to one antibody. And additionally, it is a limited resource. That means once we have received the patient's blood, we have only as much as we can get from that time point and cannot reproduce it. Another major, very major actually limitation is the scarcity and rarity of B cells in the circulation in the bloodstream of patients. It is almost like looking for a needle in a haystack, and the needle unfortunately is not as big as depicted in this picture. And I would even argue after so many years trying to find them and even finding them that I think looking for a needle in a haystack might be easier. So it's really a highly sought after goal to enrich for these B cells that are disease relevant. How do we do it? So basically, as you remember from before, I only want to have these ones that are very specific to what I'm interested in, the ones that are disease relevant, as you can see here depicted. And there are several ways of how we can enrich for the ones that we are interested in. There is a method called flow cytometry, but it's just to show you B cells do not only express the antibody on the cell surface, they have different other markers. And I can use that method to separate between different subsets and also the ones that are positive for a marker I'm interested in. And I can then use this information and only look further into the B cells that are positive that I'm interested in. Still, unfortunately, the pathogenic B cells are rather rare still within this already enriched population. So I developed so-called logs, I will call them now logs, to only specifically enrich for those that are disease relevant. And with these enriched populations at hand, we were able to generate patient-derived autoantibodies. And we were able to generate 6 of them, which is highly relevant as currently only 8 of them are published worldwide. And we could use these as an unlimited resource to characterize them and their pathogenic and functional properties. And as you remember from before, we could use their uniqueness to trace them over time. This was now more focused, this first section, on chapter 1 of the thesis. So now we will look more into chapter 2 and 3. And to understand better what was the basis of the question and mystery that we wanted to look after in chapter 2 and 3, it's important to understand what translational medicine is and what reverse translational medicine is. Translational medicine goes from the laboratory bench and takes discoveries we make here to the patient's bedside, so transform it into practical applications for patient care. In reverse translational medicine takes clinical observations from the bedside and guides scientific exploration to do that. And the clinical observation that was highly important for this thesis was the remarkable efficacy of a therapy called B-cell depleting therapy in a subset of myasthenia gravis patients. And what is B-cell depletion therapy? B-cell depletion therapy can specifically target specific B cells within all the population. And of course, you hope it also includes those that are disease relevant. And then the patient goes into a state called remission. That means he's better, he has almost no symptoms to non-symptoms and is doing well. Interestingly, these patients, after they go into the so-called remission, experience something called relapse. That means they have high disease activity again after several years. Though the question and mystery we had was, do these disease-relevant B cells survive over all of these years and re-emerge? Or are they produced de novo? Is it new B cells that are disease-relevant that develop over all these years? So we took an approach similar to Back to the Future. We tried to trace these B cells in several samples that we collected over years, over time, and again used the highly uniqueness of the antibody to try to find these B cells and antibodies. What we could find was that we could actually track one of these disease-relevant B cells over several time points in a patient, and that these B cells persisted through B-cell depletion therapy. That means it must have survived somewhere and re-emerged shortly before relapse. Thus, we could find that relapse is also associated with so-called historic old clones that survive somewhere and re-emerge during times of relapse, of new disease activity. So we now go more into the impact of this work. Of course, it is important to understand relapse in the context of patients so we can improve patient care and improve therapies. I will say that B-cell depletion therapy is already a very important tool in treatment of a subset of myasthenia gravis patients, and that all of these patients respond very well again to the same therapy. Still, we were able to identify markers on B cells that survive and persist that could be considered as additional targets for add-on therapies that might improve the outcome of patients. And additionally, we were able to show that antibody levels in this subset of disease, antibody levels rose months before the patient had any clinical symptoms. That means in this case, for these patients with this therapy, one should use or could use the antibody titer. That means the levels, the amount of antibodies in the blood for prognostic biomarker purposes. As we now approach the end of this presentation part of my thesis, I want to remind you again of the quote of Albert Einstein. The most beautiful thing we can experience is the mysterious. It is the source of all true art and science. And indeed, we looked into a lot of mysteries in this thesis. We looked into autoantibodies. We looked into ways to enrich them. We characterized them, and we tried to find out how relapse happens immunologically. So we were able to produce 6 patient-derived autoantibodies, and we could use them as very important tools to characterize their functional properties and to trace them over time. And we could see that some of these pathogenic B cells contribute to relapse after B-cell depletion therapy in myasthenia gravis. This now concludes the presentation part of my defense, and I'm very thankful for your attention.
Thank you very much for your presentation. It's now time to start with the opposition. And the opposition will be opened by the chair of the assessment committee, Professor Bos. He's professor of tumor immunology in cancer at Maastricht University Medical Center. Professor Bos.
Thank you very much, Mrs. Prorector. Dear candidate, it was a challenge to read your book. I learned a lot as a non-neurologist, and the last time I saw somebody with myasthenia gravis was actually in the lab, and it was only rats. So no experience with clinical patients, and that was in the group of Dr. De Baats 30 years ago, I think. And I'm happy that I can share some discussion with you and hope to learn even more after your nice presentation. And can we go to page 113 of your book? Because there you have figure 4, and there it already becomes very complex for me. And can you just explain me what am I looking at?
Yeah, and page 113, we were trying to understand the phenotype of B cells that survive B cell depletion therapy, and we used 10x technologies that combine the and again, like highly esteemed opponent, sorry for not starting with this.
Okay.
Yes. So we basically combined the knowledge of the B cell receptor. You can do that on the same sample, on the same cells, together with information on the transcriptomics. And there were online sources that we can use that show all of these markers that define different subsets of B cells. And we can then basically cluster each single B cell, which you can see each dot is a B cell, into different groups, like memory B cells, antibody secreting cells are the ones that are important for this thesis. And in figure B, you can see all of these cells that had clonal overlap. I will also, yes, we could see if there were clones, and if they were had also, like, the color indicates like every dot, be it black or red, is like a clone, something where there were a clonality. There was not just one B cell with that sequence, there were several. And the color indicates whether this B cell was also present before rituximab, which is B cell depletion therapy for the people still thinking about the talk, and after rituximab. And the red showed, again, I think the ones that were before. And in the last picture, we wanted to also see how was the frequency of persistent clones or persisting cells in the different compartments, especially the memory B cell subsets and the antibody secreting B cell subsets, and to see which ones are the ones where more clones persisted. And we could see there seemed to be more in the antibody secreting ones. So this whole aim of this investigation figure was to understand which are the B cells that persist, where are they mostly clustered, and is basically increased clonality, like that means having a very expanded clone where there's a lot of different B cells that come from the same origin, if that helps with survival over B cell depletion therapy. And this basically was also shown in other diseases before. And we could also see that increased clonality actually helps with survival. Basically, also, in my opinion, it's also a number of chances, because if you have more of something, it's easier to survive if you have only one cell. And I hope this could, to some extent, answer your question.
It does. Just maybe too simple for you, but you say we have a cluster called ASC. So these are the B cells that have antibodies on the membrane, and all the others do not have an antibody on the membrane?
So, no, thank you again for this very nice question. One can say that this is actually these antibody secreting cells. It is historically, it was said that it's the plasmablast and plasma cells. And plasmablasts still have the B cell receptor, which is antibody on the cell surface, and it was historically previously said that plasma cells don't, but there's new findings that show that actually plasma cells that are mainly antibody producers, that means they bring them out, that they also to some extent still express levels of antibody, that means the BCR on the cell first, but it's less. Memory B cells and the other cells express it. It's B cells develop in the bone marrow over several stages. And I think the expression of the B cell receptor antibody starts at the pro-B cell phase. And that's like when it goes through several mechanisms to make sure it does not become autoreactive and stays on the B cell, but it goes through development changes that in bone marrow can be receptor editing, which I can also explain if you want in more detail. And outside, there's a process called affinity.
I think we know that from non-Hodgkin lymphoma, how that does work.
Oh, yeah, that's true. That's true.
So these are antibody secreting cells by definition.
Yes.
Okay. And then I have a question on page 118, because then you're more interested in what type of gene expression can I see on these cells?
Yes.
And why did you look at gene expression? I mean, you want to have FACS analysis to see whether these genes leads to proteins, doesn't it?
Thank you, highly esteemed member opponent. I agree. It's FACS analysis or flow cytometry analysis gives information on the actual membrane-bound expression. And we did include in this thesis a data sets where we had this information on one of these, which is called CD20, which was important because that's the marker that is targeted with these B-cell depletion therapies. And we could see that increased levels on the mRNA level have increased levels associated in the flow cytometry.
But for me, the CD20, your interest in CD20 is conflictuous because you want to target, as I understand your story, the plasmablast, the really secreting cells, which is not the CD20 positive per se. And then I see that you have a large gene expression of BCMA, and I looked to all the figures and all the supplements, but I couldn't find is this BCMA expressed on the membrane.
So this was not investigated as part of this thesis, as we were focusing more on CD20 and its levels in that part of the thesis. I did my first research work with Edgar Meinl in Munich, where we worked on the BAFF and APRIL system, which consists also of BCMA, buffer, and TADC, and agree that this kind of therapy would be very beneficial, and also looking more into BCMA expression. BCMA is a receptor that is mainly expressed on plasma cells, and also gets that.
We know that as hematologists, we know that.
Yes,
and that's an interesting target for multiple myeloma.
I agree in there.
One more question on your antibodies that you rose, because you started your introductions that antibodies are relevant for viral infections. Do they autoantibodies express CD60?
The autoantibody, the B cells that express the autoantibodies.
Yeah, or the antibodies themselves that you find.
The antibodies themselves recognize mainly the target antigen, if that's where your question is targeted at. The B cells that produce them, we did not look into this expression of CD, what was it, CD16? CD16, no, we did not include that in our,
why do you think, as you as an immunologist, I ask about CD16? Because there is one cell that is extremely crucial in viral infections, and it's not mentioned in your thesis.
Oh,
which is the NK cells, and that's the cell triggered by antibodies via the CD16. So does CD16 and possibly NK cells play a role in the pathophysiology of your disease?
I'm sure. I think and natural killer cells are very important for the pathology of this disease. We did not look into it, but I think there's more research coming up on also other autoimmune diseases where they looked more deeper into natural killer cells and how they affect pathology. And there's also very interesting findings that actually show that natural killer cells express CD20 on their cell surface. That means targeting with rituximab does not only deplete the B cells and to some extent also T cells, but also natural killer cells. So I think you're very right. I think natural killer cells are very interesting target cell for myasthenia gravis, and disease does not only always, as like presented, of course, in diseases, more like very much focused on B cells, but the human body consists of many different cells. And I agree, I'm very sure they are very important for the immunopathology.
I think that's also cited by Einstein. Huh? If you just look where the light shines, you won't find anything that's still in the dark. Huh?
That is true. Very true.
I'm looking at the pro-rector if I can still ask one more question.
No, sorry. Your time is up.
Okay.
Oh, I'm very sorry.
Yeah. Maybe at the end.
Thank you very much. Then I'll give the word back to the pro-rector.
Thank you very much, Professor Bos. The opposition will now be continued by Professor De Baets, Professor of Chronic Inflammation in Neurodegenerative Disorders at Maastricht University.
Okay, I'm unmuted now. Okay, thank you, Mrs. Pro-Rector. Dear candidate, I would like to congratulate you with your thesis and also include the whole promotion team on both sides of the ocean. It's a very impressive thesis, but of course in the Netherlands we also like to make some remarks on the thesis, and one of them is that there is quite some repetition also in the technology when you read the thesis. I'm read it from the first page until the end, and so that's a point which could have been a bit shorter, and having all the list of techniques put together so you avoid the repetition. That said, I will come to my first question, and the first question is MuSK myasthenia is a very severe disease. I've seen several patients when it affects typically the oculobulbar region. The first patients I saw, even before we knew what MuSK was, is the patient was on the respirator and could move the arms and legs normally, which was kind of very unusual. Of course, in those days we didn't know the explanation for that. I think we now know, and Professor Punga made some contribution to that, showing that MuSK is not present to the same amount in all different muscles. But now I come to my question. You have another myasthenia, the anti-LRP4 myasthenia, which is also a bit more frequent than MuSK myasthenia, with a collaborative effort together with Socrates Tsartos, combining different countries. And what came out of that study is that patients with anti-LRP4 antibodies and LRP4 myasthenia have a much milder disease. So could you elaborate a bit on how you could explain the differences between these two forms of myasthenia, knowing that in both cases there probably is an inhibition of the interaction between LRP4 and MuSK? So that's my first question. So could you give me an answer on that, please?
Highly esteemed opponent, I think this is a highly intriguing question. As MuSK and LRP4 build a functional unit, it's like a binding co-receptor of MuSK is LRP4, and they are expressed together on the cell surface. Just for everyone to understand, an acrin binds to this and builds like this heteromer that leads to clustering of the ECHR after activation of DOX7. I think the immunopathology itself is very different. In MuSK, MG is mostly IgG4 mediated, which is very important as IgG4 mediated diseases can undergo a process called Fab-arm exchange. And Fab-arm exchange does mean nothing else than an antibody can become monovalent. And I think that is the key to your point to your answer. Mask autoantibodies can become monovalent, and then due to that inhibit this heteromer formation of LRP4 and mask receptors, whereas LRP4 receptors can still have like a dimeric structure with 2 binding sites. And we could see it when we used mask receptors in its original formation without using FABs or like Fab exchanged antibodies, that these 2 binding sites itself lead to a heteromer formation that leads to activation. So these IgG subclass that LRP4 has, which is mainly IgG1 and IgG2 to my knowledge, this cannot undergo the process of Fab exchange. So I would argue it is less strong because these antibodies are still divalent and to some extent lead to a cluster or like a heteromer formation that leads to a little bit of activation. Still not as good if there's no antibody, which we could also see in our mask antibodies, but mask antibodies have this endogenous feature that they can go through Fab exchange.
Yes, of course, we know that. I think we were actually in the Netherlands the first to publish that together with the laboratory of Genmab and Sanquin, and of course Mario Lozen and Pilar Martinez also, and ourselves contributed to that finding. Yeah, I think that's a possible explanation. I think I haven't read a good. I'm not saying that your explanations are good, but of course, why these clinical symptoms are so mild, because even a lot of clinicians and laboratories don't even believe in this anti-LRP4 myasthenia, because in some cases, of course, it's also found in other diseases like ALS, etc. So there are some doubts about the specificity of this LRP4 hypothesis. But anyway, I like your hypothesis, and it might be true, because the translation from the hypothesis in the lab to the clinics, of course, is a difficult one. My second question is, when you look at the precursor frequency of this anti-MuSK antibodies in PBMCs, how high is that? I remember publications of Antonio Lancevecchia showing that in his own blood, because the abbreviation he used for the clone source AL, Antonio Lancevecchia, and he claimed that in his measles antibody or B cells or a frequency of 1 in 112. I assume that the MuSK antibodies are B cells are much less. So my first question is, what's the precursor frequency in the peripheral blood? And do you have any idea about, is anybody looked at bone marrow cells? So please give your answer to these 2 questions, to one question.
I think this is a highly interesting question. So in one of the projects that we did not include in this thesis, I actually looked into the precursor naive cells. And the limitation was also, we did a lot of so-called bulk RNA sequencing, where we got as many B cell receptor sequences as we could out of a patient sample. We could never detect the precursor of one of our 6 monoclonals in the blood of a patient. So I would argue this is highly rare, and we were not able to do it in measles. Of course, it's easier as it's an infectious disease. Nevertheless, I do not know if anyone looked into bone marrow yet, but it is unfortunate, or it is very hard to get bone marrow. But I think it would be highly interesting, especially also given the knowledge that, first of all, also the tolerance mechanisms are impaired in myasthenia gravis, and also to see what kind of B cells are present within the bone marrow, and to understand better these naive precursors of these autoantibodies. As in the study that we did, it was highly surprising when I mutated them back as much as possible to its naive germline configuration, they still bound to the antigen, which was in that case masked with high specificity, and they were not let's say one of them was polyreactive, that means also reactive to others, and then developed reactivity towards mask very specifically, but all of the others had a high affinity already as naive precursors, as far as we could go back with what we had in hand. Let's put it like that. But it's a very interesting question. And I would personally, I don't know anyone yet. Maybe Kevin has now bone marrow. It would be very interesting. But I think it was one of the next steps to look into bone marrow and to look into spleen and other tissues to get more information on these B cells, as it was already done with, for example, the thymus that is highly relevant for AChR-MG.
Okay, thank you very much. Do I have time for a last short questions, Mr.
No,
Pro-Rector?
I'm sorry.
Okay, that's
Dank
fine.
u.
Thank you. I give the word back to the Pro-Rector.
Thank you, Professor De Baets. The next opponent is Professor Evoli, Professor of Neurology at the Catholic University of Rome in Italy. Welcome. Floor is yours.
So, dear candidate, congratulations for your great work, and I really enjoyed reading your thesis, really. And I will start immediately with my first question, that is about the first paper, the characterization of pathogenic monoclonal antibodies derived from patients. So these antibodies, two of these antibodies were IgG4, and one antibody was IgG3. And we know that non-IgG4 mask antibodies do exist. They are detected at different levels very commonly in patients. But the problem is, are they pathogenic in vivo? There is a role for these antibodies. They act as divalent antibodies. So apparently they have a role. What's your opinion on this point?
Highly esteemed opponent, this is a very interesting question that I really enjoy. So we could see, of course, there's different subclass of antibodies existing in myasthenia gravis, and most of them are IgG4. And what I could always see in every research I did and all of the work we did over the last couple of years, that it always needs like a certain threshold needs to be achieved that there's pathogenicity. So I personally think that complement or these IgG3 antibodies can also be relevant for pathogenicity in patients if they are very prevalent, and there's a lot of these there, in my opinion. Yeah, so I think it is highly interesting to look into that and how it would affect it. I would, as I said before, think because these antibodies are still divalent, say they might have a less severe cause of disease than IgG4 mediated patients that have IgG4 mediated antibodies. But one always has to say, like I said in the talk, in the blood and serum, the antibodies are very heterogeneous. There's a lot of different antibodies that interact with each other and play with each other. And it's the culmination of all of them that lead to the disease. So I'm not sure how big, if there's mainly IgG4, how big the effect of others is, and maybe if also the recognition of MuSK is a little bit reduced when the IgG4 antibodies bind to it. This could also be one of the cases that it cannot be as effective, but of course it's also important to know if you have antibodies against different subclasses, how well can they bind the antigen? Are they high binders, like the ones that we always found with our approach, or are they low affinity binders? High binders could basically take the spot on the MuSK receptor, and then the others cannot reach it, or like I do not have even the chance to reach a pathogenic capacity. But there's also other studies in other diseases that find that also these low affinity antibodies that do not interact so strongly can also be pathogenic. So that's like a highly complicated interplay of many different things that can only be understood when we have more monoclonal antibodies that we can combine and look at them integratively and try to understand the whole system. But and then we can also interplay and remove and add certain antibodies to understand this better. And I think the effect of complement would be also, in my opinion, interesting, but I'm not sure how strong, for example, that interaction would be and how relevant for the disease.
But these non-IgG4 antibodies are reduced by rituximab treatment in the same way as IgG4 antibodies. Because the evidence, is that IgG4 are reduced very much by rituximab treatment, and the IgG1 and 3 antibodies are not so much reduced. What do you think?
I think this is highly interesting, speaks again for different phenotypic phenotypes that B cells that produce different subclasses might have, in my opinion. Again, it shows that these patients, that maybe not 100% of patients respond to rituximab therapy. So one could argue now that the ones that don't respond well have different subclasses less predominant as then in their pathogenicity. And that might be the reason why they respond less. I think also that the half-life of antibodies is different, determined on subclass slightly. It's just a thought that right now came to my mind, but I don't think it's too relevant for this. That it is less affected can also be the result of different cells producing it. So, for example, plasmablasts that are very relevant for myasthenia gravis. Maybe it's mostly IgG4 plasmablasts. We could also see that plasmablasts had an increase in IgG4 actually when we did in chapter 3 look into the plasmablasts that survived or persisted after therapy. So and plasmablasts to express more CD20 than, for example, plasma cells. Okay. So these others could be from plasma cells. Yeah.
I have a question about the persistent B cells that emerge after rituximab treatment. You showed very interesting results about these cells expressing some survival markers, low level of CD20, of BAFF receptor, increased level of TACI, and of genes involved in tissue homing. My question is, these characteristics are present in these cells even before rituximab treatment, or are they selected in a way by treatment, by rituximab itself?
Could you try to give a short answer?
Okay, I will try. I think personally that they have already, to some extent, increased levels, but of course this survival that they have, or the survival benefit that they have through that, is increased also through therapy, and they will be in niches where they need BAFF and APRIL are expressed then by myeloid cells, and it then leads to this, and that's expression of more TACI, and that will lead then to IgAs, Glaswitch, for example. But I think it is both. It's always like the question, I think it's both. They are predominant and they survive through that easier, but it also, and maybe will be even more enhanced because they learn they want to survive.
Yes, thank you.
Thank you very much.
Thank you very much, Professor Ewoli. Then the opposition will be continued by Dr. Hoeimakers, neurologist at the Department of Neurology at Maastricht University Medical Center.
Thank you, Mrs. de Pro-Rector. Dear candidates, first I would like to congratulate you on this wonderful thesis, and also the congratulations for your supervising team. As a clinician who frequently sees patients suffering from myasthenia gravis, I was glad to read the new insights on my anti-MuSK-MG. And although I'm familiar with the general pathogenic mechanisms, some parts of your thesis about the detailed laboratory experiments were quite difficult for me to understand. But I was happy that you had great illustration, and also so your lay talk was really helpful for me. I would like to discuss some possible clinical implications of your results, and I want to go to publication 3. And in publication 3, you have found that clonal variants of a pathogenic MuSK-specific B cell were identified prior to post-rituximab relapse, together with increased MuSK autoantibody levels. And you state that both may serve as a valuable prognostic biomarker, for predicting the relapse. And I was wondering, is it necessary to check both, or can you choose one for that?
Thank you, highly esteemed opponent, for this very important question. Yes, one can only check for one, and of course the antibody levels is easier and also much cheaper than checking for the pathogenic B cell. As you might remember from the talk, it's really hard to detect these antigen disease relevant B cells, and I did get these by sheer force and using everything we have in our biorepository. And maybe in the future, these approaches will be easier, and I had to develop this bait antigen to isolate them. But in the clinical practice, I really would recommend we could even see it already 7 to 8 months before clinical detectable relapse in patients, that these titers start to increase. And 2 months before, they already have the same high level than they have 2 months later when the patient has relapsed. That again showed to me that there's a threshold effect that needs to be reached for pathogenic capacity. And I would really recommend in these patients to measure it, so you can treat them and give another circle of rituximab before they get into relapse. And I think that would help, and that also works really well in patients. Maybe even using, because ocrelizumab is also a B-cell depletion therapy that works on slightly more, even still more on plasma cells, and is a little bit more expressed. I would even also say that would be even a better candidate therapy, in my opinion. Just like, but rituximab is also very, very good. Yeah.
Yeah, and I think you also illustrated that with your tables and your figures in your thesis. And yeah, you already told me that it's more feasible to check the autoantibody levels than the pathogenic B cell clones. So I hope that you will develop an easier test for that, because it sounds promising. And yeah, already in 2019, Triplets also suggested to check to monitor the MuSK autoantibody levels. And based on your findings, which frequency of monitoring do you suggest?
I think because patients always experience relapse after 1.5 to 3.5 years, that was always what we had, at least in the one has again to say there's a very rare disease, and we have maybe only like a limited view on that by all our patients at Yale, and maybe also like where we live and what kind of patients come. But like that was always the frequency we could see. So I would say like after 1 year, I would start checking every half year.
Every half year.
Yeah, I think.
Yeah,
I think that would be a good start, and and easy to do. One of course always needs to ask the laboratory which measure assays they use. Do they use radioimmunoassay, ELISA, or cell-based assay? I would recommend trying to find one that uses a cell-based assay, as it's more sensitive and better, in my opinion, to check. Yeah, that's also like something that one always has to consider when to measure it, and to figure out what kind of assay is used by the clinical practice. And for example, we had patient data from the clinic for these samples, and I used my own cell-based assay again, and it was very nice that most of the titers were the same, that I could also see over time in these patients, but every half year, and also try to understand what assay is used.
I think that will be feasible.
Yeah.
And you have used the MG composite score to measure the clinical deterioration in your patients. But in anti-MuSK MG, especially the facial bulbar muscles are involved. And is the MG composite sensitive enough to measure deterioration of these complaints, or do you need another outcome measure? What do you suggest?
I have to admit that I studied medicine, so I'm an MD, but never worked as a medical doctor, and got the MGC through a collaboration with Richard Novak. So I would need to really read up what goes into that score, but there's different other scores which also include that. I forgot how that one was called. I recently wrote a sort MGIDL, like how the daily living is affected. So I think basically we always use also the MGFA, which is more like the severity of this disease and things also affected. I think it's a lot of different scores that should be measured to get a complete picture, but I focused here on the MGC MGC just for the purposes. It gives you a number, and this gives you like a scale like from 1 to something, but it really gives you a number. It's not like the MGFA where you have like 1 to 5. It gives you more acceptable numbers, and it's much easier then to correlate it with titer and to look into different effects when I show it. But I agree with you for this, one needs to look more into all the different ones and if they can measure more what is mass specific. But
Yeah.
you're more the expert in that than me. Yeah.
Well, I agree that the suggestion of the MG-ADL is really good. It is used nowadays in a lot of trials. So I think that's a really good suggestion. And well, you say, well, when you use the biomarkers for predicting the post-rituximab relapse, at what point would you restart the treatment? Because you want to prevent the relapse.
Yeah, I think in Germany, I talked to people when I gave a talk in Munich. They give it, I think, at certain time points, maybe like every year. But I personally think also it's a highly effective therapy. It still has side effects. So I would use the approach of measuring the titers.
Mm-hmm.
And then when I can already detect a titer, I would treat the patient again because it works rather fast. So to not to have the patients go to maybe unnecessary titer because he or she is someone who would experience new relapse in 3 years, but then we already have like 3 more cycles that we gave to them. But that's just my personal opinion. Maybe it's easier and easier plannable if one does like for all the patients the same interval, but personally, scientifically, immunologically, I think one could look into the heterogeneity by measuring the titer.
But is there a cutoff value for the titer?
There are cutoff values for the titer, and one does it always like yeah, there's a cutoff value that you can see. You always have, and then when you run the assay, you have a highly positive control and one that is slightly positive, but still positive, and one that is negative control. And if it's between this low positive and high positive control, as the CBA is, for example, run at Oxford University, then you know that patient has antibodies, and the titer is just determined on how many dilutions one does. Like dilution is like making things less concentrated by going down and always taking the same amount again, and looking until that dilution I still can over the threshold or this low affinity or like low interaction that is positive.
Yeah. Okay. Thank you for your answers, and I will give back the word to the prorector.
Thank you very much.
Thank you very much, Dr. Hoemakers. It's now time for the last opponent, Professor Punga, Professor of Clinical Neurophysiology at Uppsala University in Sweden.
Thank you, Mrs. Prorector. So, dear candidate, it was a pleasure reading your thesis book. So, one of the most important research questions and problems is the lack of objective biomarkers in myasthenia. So, would your findings of clonal variants of these MuSK antibodies help further develop these MG-specific biomarkers for disease progression, you think?
Thank you, highly esteemed point for this interesting question. And I know that you are the expert on biomarkers, so I'm not inferring that I'm the super expert in this. I think the clonality is very interesting, and in my opinion, by having so little every time we try to find clonal variants, sometimes at the same point we found clonal variants of the same clones, which suggested to me that maybe also it is a very polyclonal disease as well. It's less polyclonal than one might think, especially in the ones that are highly pathogenic or high binders, as we call them. So I think only looking for the ones, one could narrow it down, the titer, to the ones that are clonal, but there's currently like no perfect and easy way to measure it, which could be implemented into clinical practice. That would not be very easy. One would need to try to like get the antibodies out of the blood and then try to separate them somehow from each other with maybe different properties of which epitope on MuSK they bind would be like maybe one suggestion. Then one would need to cut it and do mass spectrometry spectrum, which
Yeah.
like
And you
it
see,
would
this is not feasible, right?
No,
for the
it's
clinical
not.
trial. So
No.
what is required, you think, in order to get there, to have something like a quick kit that you can measure the polyclonality very easily, or the
What I really like recently, there's very cool essays coming out. Sorry for using the word cool. I just like them. It's
No,
like
it's fine.
these immunodot essays where they add basically something on a sheet of paper like this, different dots of a protein, and you can within 3 hours determine if it binds or not with negative and positive controls. And for example, one could use that from different MuSK epitopes and just add different sections of the MuSK protein, express it and put on the dot, and that would, for example, work really fast. And then you can say, hey, there's a lot of these that mainly detect the Ig like domain 1 or Ig like domain 2 in these patients. And one can also then determine if are they more IgG1 subclass 2 or 3 and 4. Like there's a lot of different detection steps you can add, but it makes it highly easy to reproduce and use, and it can prepare. And I think in a recent study I read, they just prepare these sheets and store them for like half a year or even a year, and it can be used. Like this would be possible, but one of course needs to add all the right controls to make sure that what you see is correct. One needs to validate that the subclass antibodies are correct, because I encountered a lot of these antibodies that are not specific for the IgG subclass that they are sold to be specific for.
So I would encourage you to go into that and to help in the biomarker field and validate these ones. That sounds good. So then I wonder, because I work a lot with the pathophysiology of MuSK myasthenia as well. So what would you consider the optimal preclinical model to study the pathophysiology of MuSK myasthenia, and why?
So preclinical model outside of animal or within animals?
Within animal, but not human.
Within animal. I think, for example, because the sequence homology of the MuSK receptor is very close between mice and humans, that mouse is a perfect candidate for investigating the pathogenicity of MuSK autoantibody, and has already been very successfully used by Margie Hübers in her laboratory in MuSK monoclonal antibodies. Additionally, I think personally, pre-human or like outside of human, we have also like the C2C self-assay that you can use outside, that is also mouse-based, but there's also interestingly human organoids that, in my opinion, in the future can be used to test this in a more related human way that would spare the life of animals. But these are the different preclinical ways I would right now think are the best. But like C2C12 that I used for the thesis is really already a good approximation and can already show very well the interaction of MuSK and LRP4 and how it inhibits the AChR clustering.
Would you consider that they represent human muscles well, the C2C12?
Of course, there's a species difference, but it does, I think, in the functionality. I cannot speak for the whole of the muscle because I did not do extensive sequencing and compare between these. But I think for the functionality of the MuSK LRP4 pathway, and in that regard, like the association with acrin, I think C2C12 is a very good assay.
Yeah, that's good. You know the limitations as well. So the last question, based on your data, what do you think would be the most promising treatment for disease remission in MuSK antibody MG in the future? And you may not say rituximab, or you may think outside of rituximab as well. Is there any other treatment you would think has the highest promise of being the treatment for MuSK myasthenia, maybe myasthenia in general, to achieve remission?
CAR T-cell therapy, which in which this is a therapy in all other B-cell depletion therapies. You target also a lot of innocent B cells that would actually be good to have and not to lose. There's new therapies called CAR T-cell that specifically target only the B cells that are specific for example, MuSK or AChR, which is the other subtype of disease. I think this is a highly promising approach. The only question for me in this therapy that still remains is how good can it go into tissues? Where are the cells located? Can it eliminate them? And is it or like how often do you have to give it again because cells re-emerge from like a niche that they were in. But I think that's for me, the most promising one, the ones with where I expect the least amount of side effects. But I think also currently is a very nice time for patients because we have also other new therapies, including FKTG-MOD, where we can reduce antibodies that are already present right now that work really well. And I still think rituximab is highly effective and very good. But there were also recent studies that looked into ocrelizumab, which is anti-CD19. When you look at B cells over the development, one could like say, like, if it's like this and this is the thing, rituximab attacks the ones in between my hands like this, and ocrelizumab goes a little bit farther. But we are here right now interested in the ones that produce antibodies, and we get more, we remove more that have like antibody secreting function. So that could also be good, but have potentially, of course, more side effects.
Okay, thank you. I think my time is up.
Oh, sorry for always
Or thank you very much.
Of course.
Podcast. Orator, you're muted.
Am I muted?
Now we can hear you.
Okay. Miriam Fichtner, again. The time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and of your defense. I request that you and your company await the results of our deliberations and our return. Fichtner. The degree committee here present online has assessed the quality of your thesis and your defense in view of its positive verdict and taking into account your previous qualifications. The degree committee has decided to grant you the degree of doctor. Professor Martinez is authorized to confer upon you this academic distinction in accordance with Dutch University custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I do.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Miriam Francisca Laura Fichtner, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor affixed with the official seal of the university, signed by the beadle. And I would like now to give the floor to Professor O'Connor, who will say some words about you.
Okay, thank you very much. I just wanted to say that it was my sincere pleasure to have Miriam working in my lab. She was here at Yale for 5 years working with me, and the work that she presented today is the product of that. So for those of you who may not have visited Yale before, it's very close to New York City. And when Miriam was here, she took advantage of being near New York. She often attended the US Open tennis tournaments that took place in the fall. She very often went to Broadway shows in New York. She saw almost 50 Broadway shows while she was here. She enjoyed also traveling to New York during the holiday season because the Lindt store in New York City is one of the only places in the world where you can get a very unique Advent calendar, which she collected. And then in the holiday season, there was also the baking of German cookies and treats, which were gladly received and shared with all of us. And then next week, Halloween in the US was one of her favorite holidays as well. She's a big Tim Burton fan and enjoys Nightmare Before Christmas. And then I wanted to tell a short anecdote about the way that Miriam worked. Miriam's commitment to work and her dedication and her hard work were completely unwavering. And I think it goes without saying that she worked hard, because you've seen the amount of work that she did in the short period of time that she was here. But a couple stories to really highlight that. When the pandemic started, Yale moved very quickly to get the labs back open. And the way that it worked was that everybody could sign up for a bay to do their lab work. And a lot of people were reluctant to come in, but not Miriam. She signed up for bays as soon as they were available. And if anybody didn't sign up for any other bays to do their work, Miriam signed up for those bays too, and continued to work through the pandemic while still being safe, masked and socially distanced, and got a lot of work done. It didn't end there. There was a couple other periods of time where Miriam demonstrated that nothing would get in the way of her work. I got a call on a Saturday morning one time that she told me that there were people in the building who were cleaning the floors in the lab, and they told her that she had to leave. And she called me and asked me to tell them that they can't tell her to leave, that she was doing experiments. And so she negotiated with them to work around her. And so they cleaned the floor in one part of the lab and then left her for another. And then finally, this last story, I think, really demonstrates her commitment to work. And again, not letting anything get in the way of her hard work. There was a suspected, and I emphasize the word suspected, gas leak in our area here. And they evacuated. Miriam's laughing because she remembers this. She didn't know I was going to tell this story. They closed all the streets around our building, and they closed our building. And again, Miriam called me on my mobile and said that She had cells that needed to be fed, and she needed to get all of her antibodies out so she could purify them, because they were telling us that they weren't going to open the building or the streets until they had found out what was going on. So later that afternoon, it was early evening, she called me and said that the cells had been fed and the antibodies had been rescued and I can purify them. And I said, oh, did they open the building? And she said, no, they didn't guard the back door of the building. So I went in the back door and ran up to the lab through the back stairwell and fed my cells and took the antibodies to another lab to finish the work. All was well. It was not a gas leak. Somebody had spilled mercaptoethanol, and it just smelled like a gas leak, and it never was. And so a lot of work was interrupted, but not Miriam's. So, and you know, all of this hard work and dedication you saw today, but some numbers that are amazing is that while she was with me, she published 10 papers, some of which, one of them was in the Journal of Experimental Medicine, Journal of Immunology, and Acta Neuropathologica. So not only a good number of papers, but good papers in good journals. And she got more awards than any postdoc that's worked with me. She won a Brown Cox Fellowship, which supported her while she was here, a SPIN research grant, and a DFG grant from the German government as well. So this stands out in my view of what really hard work looks like, and is an essential ingredient for a successful scientist. And so Miriam, I hope you continue to not let anything get in the way of your hard work. And I'm sure that with that, your drive and determination, you'll continue to be very, very successful. So congratulations.
Yes, I would like Miriam also to congratulate you for the very nice thesis, but also for your commitment, as Kevin has mentioned, and enthusiasm every time we discuss, always very enthusiastic about science and always very ready for discussing. I really would like also to give the congratulations to your family. I don't know if they could attend via the online link, but I would like to congratulate them. And yeah, and give my compliments for the very hard work that you have performed during your PhD, and you deserve the PhD title. Mario, would you like to say something as well?
Not really all has been said, but I would like to thank Miriam and the collaboration team at Yale for these very fruitful work that we've done together. And I wish you all the best for the future, Miriam.
Thank you very much to everyone. One does not know what to say when one has been showered with so many, first of all, so funny, but also nice compliments. So I want to thank everyone again who was involved, and all of my family and friends who always supported me. And I always enjoyed scientific discussion. So I also really enjoyed talking today. And if anyone still wants to talk, I'm always one email away and happy to discuss science, because it's fun, and that's how we get new ideas and improve our ways. So thank you very much for everyone, Pilar and Mario, for all of the great collaborations that we had over the year, and Kevin for being such a great mentor, especially over the last couple of years. I want to thank everyone, and also University of Maastricht for awarding me this honor today of the PhD. So thank you very much. And also to all of the committee for taking all the work to read through this thesis, which I know it's like kind of big, but I hope the beautiful surface helped made it a little bit easier to read through it. Thank you very much.
Dear Dr. Fichtner, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. Also, congratulations to your family, hopefully online. And congratulations, of course, to your supervisors. And I'd like to thank all the members of the degree committee for the work they did for this PhD defense and their presence here online. Special thanks to our guests from outside Maastricht, Professor Evoli and Professor Ponga. Thank you very much for being here. Your input is very much appreciated by Maastricht University. Dr. Fichtner, I can tell you that the degree committee was very happy with the way you defended your thesis. They were especially impressed by some of your answers that really showed that you have great insight into the topic. I personally enjoyed your enthusiasm during your defense. And it's also, I think, very, well, maybe it gives some impression of you that you're in the lab, apparently defending your thesis. I don't think that happens very often. Usually people are at home or in a quiet area, but you're in the lab. So probably that also demonstrates that you're really enthusiastic about the research you've been doing. I hope you enjoyed the ceremony. Well, you said you did, so that's okay. But now it's time to relax and to enjoy the rest of the day and maybe celebrate the fact that you have obtained the doctorate from this university. And with this, I will close this academic ceremony.